Okay, here we go for the last session of the 2025 Immersion project presentations. We're in the middle of session 7, session around species, behavior, and ecological interactions, where we just had one presentation that was confidential with Gaël and Daniel, but with the students, presentation that was around behavioral analysis of the visual motor response of cephalopods. Well, we were talking about octopus, and we are going to that at the end with the last presentation with Raphael Tremblay. And before that, we will have Andrea Vera talking about Mustelus, and Jacob Rockel talking about turtles and crabs, also somewhere in a really warm ocean. In this jury, we have Aziz Mula, postdoc on coastal ecosystems here at the University of Côte d'Azur, Dominique Adriens, online researcher at Ghent University, Stina Kolodzej, postdoc at Aarhus University, and Juliette Armeni, project manager at Polmer. Thank you for being with us. Also joined for the reviewing of the papers. Also have Camille Boucaud of this work. Andrea, do you have a 15-minute timer making noise also at the end? So if you're ready, enjoy the comment.
Perfect. When I say the word shark research, you may think about diving and big sharks, right? Well, today I'm going to take you to a different setting, fishing ports and baby sharks, which is often an approach that is often overlooked when thinking about shark conservation. This is why during my internship with ISD, I decided to study the spatiotemporal distribution of the different life stages of the commonest bloodhound Mustelus mustelus, and the interaction with the fisheries in the Immersion Gulf in Greece. And this research is particularly interesting and relevant because elasmobranchs, so shark rays and chimeras, they're the second most threatened vertebrate group worldwide, which brings the importance of protecting their nursery habitats in order to promote the population's recovery. So taking this into consideration, in Greece, Greece hosts 4 important areas for sharks and rays, so-called the Isras. And indeed, the Immersion Gulf has already been recognized as an important area for 3 threatened elasmobranch species, mainly for their reproduction. Among them, we find the commonest bloodhound Mustelus mustelus, which is found endangered globally and locally in Greece. Also in the Mediterranean, its population has decreased between 50 to 79% in the last 3 generations. And due to its habitat use, it's the demersal shark that is mainly found in muddy and sandy grounds. It feeds on crustaceans, mainly shrimp and mantis shrimp. It's very vulnerable to different types of fisheries. And in more locally, in the Immersion Gulf, there has already been seen, as you can see in the pictures, evidence of immature individuals concentrated and interacting with fisheries. This is why I have these 4 study objectives. First is to map their distribution around the Gulf in terms of spatially and temporally. Then look at the essential habitats for the early life stages. Look at the interaction with the fisheries, and taking all this information into consideration, providing management recommendations for the conservation of this species in the Gulf. So let's dive into where I was and what I found. The Immersion Gulf is this very small gulf in the western side of Greece, in the Ionian Gulf. It's around 405 kilometers. It's a national park. It has 2 Natura 2000 sites. It's a key biodiversity area, and it's, well, among other recognitions, but it's also found in the Bonn, Bern, and Barcelona Convention. So its ecological importance and biological importance has already been recognized. Now, into the fisheries context, only a small-scale fisheries operate in the area, with around, well, minimum 150 commercial fisheries. And if you can see in the map, there are mainly 8 fishing ports around the Gulf. Now, from fishery-dependent data gathered around the Gulf from 2022 to 2025 under the Bioelasmo Catch project from IC, we were able to get 1,247 fishing trip surveys, which the ones I participated in, 74 of them, from April until July. These surveys were done at the port and on board to the fishers, in which we gathered fishing trip data such as the environmental data from the fishing trips, also the GPS location, the depth that they were fishing, the gear that they use, and all the specificities of the gear they use. And we also gathered biological data of the individuals captured. In this case, from the sharks, we measured the total length, we took the sex, the maturity status, so they were immature or mature. We also weighted them in case that they were dead, and we took stomach and DNA samples. We also did a at vessel and short-term post-release mortality assessments in order to assess their health related to the fishing activities. I will not go into details of these in my presentation, but questions are open if you want to know more about this. Then, in order to analyze the data regarding the live stages, I created this classification that is an adapted version from Cyprian et al. With all the information that is from this species worldwide and locally about their maturity and their different life stages. And taking into account their sizes, but as well some physical cues, such as the presence of the umbilical scar that you can see here, or the level of calcification of the claspers for the maturity of the males. Now, for the data, I ran some distribution maps with QGIS, and I performed different non-parametrical tests in RStudio, as my data was either categorical or like homoceticity and normality. I also did a CKUE standardization for the number of sharks per kilometer of net per hour, as well as the number of sharks per 1,000 hooks per hour. So let's see what I found. In terms of the spatiotemporal distribution of the species, from the 1,247 fishing trips in this period, 107 of them captured Mustelus mustelus, with a total number of 326 individuals captured. And if you look closely in the map, in the central north area of the Gulf, Coronesia, the fishing trips landing in Coronesia accounted for 88% of all the catches of this individual. And if you can see, it also overlaps with a high fishing activity zone. And from these 326 individuals, 88% of them were either newborns and young of the year, which means that they were individuals smaller than 55 centimeters. And if you also look into the map, we had some adults that they were captured, but their distribution was more peripheral, and some of them in the central north area, but also around other areas of the Gulf. So now into seasonality and depth, we saw significant differences among life stages. Newborns were using mainly shallow depths between 5 to 7 meters, and they were usually present here in late spring. Meanwhile, young of the year and juveniles were mainly present in summer, and young of the year juveniles in late fall as well, in early fall, sorry. And adults, they were present from February until September. And we had several encounters with pregnant females. As you can see, they were found in February and in June. And these three life stages, they use different depths compared to newborns. And these findings, in terms of depth and season, they align with other popping behaviors that have been seen in the Canary Islands, in the Ligurian Sea, and in an NP8 in South Africa, in which females take a birth at these very shallow areas. And our findings also suggest that there might be an ontogenic habitat shift within the area. So now, I want you to remember this concept, métiers. A métier is a specific combination of a gear type, a targeted catch, and specific parameters of depth and time of fishing. In Ambracticos, we can find three main métiers that we can group them by the gear they use. With travel nets, we find Gambarodicta métier, which targeted shrimps. Then we find Tiara, that targeted paddlefish and flatfish. And then we have with long lines and gill nets Esquilodicta, that directly targeted Mustelus mustelus. And if you can also see, they vary in range of depth and fishing time as well. So now, in terms of fisheries interaction, we saw a very strong material-specific patterns. 82.5% of all the catches were with Gambarodicta metier. And from these catches, 94% of them were from newborns and young of the years. And if you can see all the gears that they use, trammel nets that are the GTR, they mainly capture newborns and young of the year. But if we look into Esquilodicta, using gill nets, only 11% of the fishing trips were using these metier. And they mainly captured adults. And from these adults, I want to highlight that 6 of these individuals were pregnant females. And this material also presented very high mortality, with 79% of the individuals already dead when they reached the boat, and 100% of them dead at the end of the fishing activity. And this actually shows that specific materials and gears disproportionately affect different life stages. Trammel nets mainly targeting early life stages, meanwhile, gill nets target mainly adults, mainly with Esquilodicta metier. And this pattern has also been seen in the Gulf of Caves with the trammel nets. Finally, in terms of catch per unit effort patterns, we saw the same thing. Coronesia, this central north area of the Gulf, the fishing trips there accounted for the highest mean CPUE and the highest maximum CPUE of 16 sharks per kilometer of net per hour. Yet, we also saw some ports with 0 catches or interaction with sharks, or with very, very low CPUE. And this takes me to my management recommendations. The first one is to have seasonal and spatial closures in these high areas that overlap with newborns young of the years, mainly Coronesia, and identifying suitable alternatives for fishing grounds during this period. Then, gear modification, mainly for the trammel nets, and implementing bycatch reduction devices like the ones that are implementing now with the Life Prometheus project using magnets and in a near future LED lights. Finally, banning the targeted catch with gill nets and long lines, so with Esquilodicta metier, as there is already the history of the local extinction of the sandbar shark, the Carcharinus plumbeus, already in the region. And this highlights that it's imperative to take action to prevent this happening to this species. And finally, expanding fishery-independent efforts with BROPs that we have also been doing in the Gulf. So finally, I have these 2 take-home messages. Minimizing fishing pressures is key in key reproductive areas, is essential to maintain population stability. And also working and collaborating with local fishers, mainly to improve their handling practices, is very important for them as well to support sustainable shark conservation. Finally, I want to thank you all for your attention. I want to thank Maris to allow me to make this internship. Martina Cifren, my supervisor, that she was very, very supportive all the way, and we share this passion for Mustelus. And IC for allowing me to attend the EA conference and present this work as well. And I just wanted to leave you with this final message. Nursery areas are the heart of regeneration and survival of the species. So it's imperative that we protect them while working closely with local communities. Thank you.
So with the last presentation, we ended with you. Let's start with you this time.
Very good. Congratulations. So much information. Oh. But you did a lot, right? It's very impressive. I do want to know more about the AVM, if possible. How was this determined? And you mentioned there was a excess mortality in the adults at the end. Why do you think that was compared to the?
Mainly because the adults, they're targeted catch. So they're targeting them, and let me go here to the materials. Okay, here. So also you can see the fishing time with the Gambarodicta, with the Esquilodicta, but they're targeting Mustelus. They leave the nets and the hooks between 10 to 24 hours. So that's why, first of all, they add vessel mortality. So when they reach the boat, they're already dead. And then they also have commercial value. So they always retain it. They never let it live. So they always retain the shark. And that's why we have 100% of post-fishing activity mortality.
Okay.
Yeah.
Are these, I'm going to ask a really stupid question.
No.
Are these sharks being consumed?
Yeah.
Yes.
Yeah. They consume them locally, the big sharks. The small ones, they don't have any commercial value. So that's why most of them, like they give them to us, or they just release them while they're like are cleaning the nets. They just release them because they don't have any commercial value. But the big sharks, they consume it there.
Okay. And a final question on working with fishers and speaking to them. Do they agree with your management recommendations? Are they supportive of them?
Yeah. Okay. Well, of course, I was in Greece, so the language barrier was big. However, I can say with the few fishers that I was able to speak, and then looking how Martina, she was the main one interacting with them. She speaks Greek, and she has already been there for like 3 years. And one, they are lovely people. Like they're super open to hear you, and something also very interesting is that now the age of the fishermen is starting to be like to get older and older and older because nobody wants to become fisher there in the Gulf. So they are open to know how to improve the fisheries, and because they have seen the effects of the mismanagement and not taking care of this species. And in terms of these management recommendations, with the Life Prometheus project, they're already working with the local community, with the local fishers. So they're placing these magnets in their nets to prove and to see how much and the degree that it reduces the bycatch there. So they're very open because they're not targeting them, mainly with Gambaro di Tanchiara. Yes.
Very good. Well done. Thank you very much.
Thank you.
For you? Yeah. Let's go to you, maybe, Dominic. Go backwards.
Okay. Thank you. Very nice results, and also very important results related to the species. So you mentioned that the shark is caught both as bycatch as well as from targeted fisheries. Do you have any idea in your data set to what degree there is a potential bias, in the sense that in the size distribution of the ones that were caught and the number of specimens, if you would compare targeted fisheries compared to bycatch? Is, for example, bycatch only the smaller ones and targeted fisheries is the larger ones, or does this also impact the smaller ones?
No. Yeah. For example, here, if you see newborns, young of the year, juveniles, and adults, and the answer is actually yes. So, bycatch is mainly it's mainly impacting young of the years and newborns, so very small individuals. And targeted catch is mainly affecting juveniles and adults. Indeed, with the targeted catch, there was never any catch of small individuals.
Yeah, and I see on the graph, it's not only the proportion, but also the absolute number. The total number for the smaller ones is way larger than for the larger ones. And so at the population level, that has a huge impact.
Yeah.
So I was wondering, because it all seems indeed that this Gulf is a nursery area for the shark species. But do you have any idea what the fishery output is of that species outside of the Gulf, like in the Mediterranean? If they do similar kind of fishing techniques, is then the proportion of, for example, small individuals versus larger completely different, which would then indeed be a confirmation that the Gulf is a very important nursery source, which is not taking place in other parts of the Mediterranean?
Yeah. Well, in terms of what I presented, for example, the Metiers, this is very not only species specific, but area specific. So Gambarodicta, Chiara, Spilodicta, they're very specific for umbrachicals. But the gears, travel nets, and as I presented before, there are, for example, in the Gulf of Gibbs, they also saw this pattern, that travel nets, they mainly bycatch small individuals. And also in other areas, I know in the Canary Islands, they also saw like big individuals interact a lot with these type of fisheries. Yeah, and also something to take into account. This is something that we learned recently. Like Martina told me recently, the population of Mustelus inside the Gulf is highly isolated from the rest of the populations outside of the Gulf. So this even highlights the importance of the Gulf for them as a reproductive area and for their survival.
Okay, so that means once they leave the nursery, they still have to undergo some kind of migration to the place where they mature and spawn and so on.
Yeah. I would say umbrachicals also is not only the nursery, but it's also the pupping area for these sharks. Because we have seen pregnant females with already developed pup inside. Actually, one of the sharks, the pregnant females that was caught, she had already like 15 pups inside, that they were almost the size at birth. So yeah, that really highlights that we don't know if they go out. That is something that we haven't tested. We haven't put any satellite tags, which would be really good to know if they leave and then they come back to put their pups. But yes, this area is not only a nursery, but it's also the pupping area of this shark.
Yeah, and then maybe a last one. Based on the fishery data you got, do you know whether this Gulf is also nursery ground for other fish or other important species?
It's actually a nursery as well for other two threatened rays, the duckbill eagle ray and the butterflies. Yeah, great. Yeah. For two other rays, and they're actually, which is interesting. These two rays it's prohibited to capture them, and you need to release them alive. But for Mustelus, it's not this. But there is and we have also seen for torpedo. When we were doing the surveys and everything, we had a lot of cases when I was there. A lot of pregnant females that they just release the babies due to the stress for fishing. So it might also be a nursery ground for many other ray species.
Okay. Thank you very much.
You're welcome.
Thank you, Dominique. We can come back to Stina.
Yeah, thank you for this really, really cool presentation. Most of my questions have already been asked, so I will ask, in the beginning, in your introduction, you said that the population is declining in the last 3 generations. Can you just define for me what 3 generations means?
Well, they have like a generational length of, I think it's like 15 years, I think, more or less. So it's in the Mediterranean, so then it would be in the last 45 years.
Okay, so when you talk about a generation, you mean a shark's generation and not...
Yeah, shark generation, yeah.
Okay. And you also talked about the life stage classification. Maybe you can go back to that slide.
Yeah, I have it here, more detail.
Yeah. So did you come up with that for the population that you looked at in the Gulf?
Okay, so this, as I mentioned, this is adapted from Cyprian et al. So, which is my supervisor. They're the ones doing the work in the Gulf. This is a bit more tailored. I did it more tailored to also the numbers that we have in the Gulf. So before we had size at birth, that was a bit higher. And when I was there, we encountered some newborns that were smaller. So I kind of tailored that to reach the actual newborn sizes that we were seeing. And I added the classification of young of the years that was already set by the shark specialist group when assessing the Umbrachicos Gulf as an ISRA. So I just kind of put everything together. And as we, in the field, we We saw if they have the umbilical scar. I use that also as an additional clue to classify individuals as newborns. So I did a bunch of like calculations between all the sizes and characteristics in order to obtain the final classification of the individuals. Yeah.
But that's really cool. So this is for you. You collected this data for the Gulf,
Mm-hmm.
and the supervisor collected the data globally or for the Mediterranean?
No, my supervisor, they do. So I see they have the project that is called the Biological Catch Project. And it's focused in the Mediterranean, but mainly in Greece. And this project is focused in umbraticals right now. So they study the rays and the sharks in the Gulf and their interaction with the fisheries. So the global data that I used was from other papers and other publications, but the local data that I used was from all this historical data from 2022 that has of the captures of the individuals. Yeah.
But that's really interesting. That means that your population in the Gulf has like there's very small or it's very distinct in its life history traits compared to other populations in the Mediterranean.
It's yeah, like I saw the size at birth, it was a bit higher, and here I saw it a bit smaller. The rest, honestly, there's no information about the young of the years in other sites that I checked. Mainly people only focus in the size of maturity and when they're born. But for me, it was important to make this classification because it's different that you say it's an individual of 42 centimeters or 55 centimeters than an individual of 100 that is still a juvenile. So, yeah.
But if you see that they are so in your Gulf that they are relatively distinct from populations in the Mediterranean in general, what do you think does it mean about mixing of populations and movement?
I honestly, I would say only when they're juveniles, they might be going out of the Gulf. I don't think they will live that much. Also because we have encounters of like young individuals, like the not already mature individuals, but already like big sharks, but not only like the mature ones, like only the reproducing animals. So I may think they might live, but they come back. And that is also a pattern that has been seen with the rays there, that they are very, very localized within the Gulf, and their populations are very isolated. So this kind of makes them even more vulnerable to the pressures that are inside of the Gulf. Yeah.
And again, one of my favorite questions, working with live animals, working with charismatic megafauna, even though these sharks are not really big. What about ethics on this?
Well, I would say the thing that we mainly questioned ourselves was what to do with the dead animals. Like when they already were dead, with the fishing practices. Because we took the stomach samples, for the rays we took the spine, and then we took DNA samples, but then of course you have the whole body. And IC has some very strict policies in animal consumption and everything. So we mainly, if there were cats there, because in Greece and in the Gulf that they were there, we tend to give them the meat to the cats that they were there. But these animals, they were just thrown away, back to the sea. So I think that would be like the main thing, like the main ethical thing. And then they were bycaught. So I wouldn't say it's like people they're catching it for us to study them. So I wouldn't say there's this problem when studying them in the fisheries. Yeah.
And you said that there's a targeted fishery for the shark. Yeah. So how was it easy, or how was it to establish the collaboration with the fishermen? If this is a targeted fishery, so this is their livelihood, were they willing to share, or was it difficult to work with them?
Yeah, honestly, Martina, when I was there, I was never in one of the targeted vessels, targeted fishing vessels. But Martina, as I mentioned, she has already been there like 3 years. So she has built this trust from the fishing community. And it's like, yeah, she has told me at the beginning it was hard because, of course, like they don't want to tell you, but at the end, for them, they're not doing anything wrong, and it's not prohibited. Like it's not something prohibited and something wrong they're doing. So also, like, why would they hide this from you if it's a legal catch? So yeah, I would say it was a process that took time for Martina, but I think now fishers, they like you can see the trust they have on her. Yeah.
Okay, great. Thank you so much.
Yeah.
Thank you, Stina. We'll finish with you, Juliette. It's not always easy to finish after so many questions, but I'm sure you have more.
Yes, I do. Thank you very much, Andrea. And it's very good to know that cats are eating sharky in Greece. But
Anyways.
I'm not entirely clear about the organization when you did your internship. So maybe can you just present it a bit, and also
Yeah,
the
sure.
funding, how did you it, and so on. It was just the introduction, but I think it's really important to get it. And yeah.
Okay, perfect. Thanks. So yes, I did it with IC. IC is an NGO focused in the conservation of elasmobranchs in the Mediterranean, mainly in Greece, and they have different projects in different parts of the Gulf and different islands. And my specific project is called Bioelasmocatch Project, and it's actually part of the PhD of Martina, my supervisor. So in this project, a Bioelasmocatch project focused in the interaction and the biology of all the elasmobranchs. So not only my species, but all the elasmobranchs and all the rays that are found in the Gulf, and see their interaction with the fisheries in order to exactly reduce the bycatch and see measures that are possible to apply in the Gulf. So yes, they're studying, well, we also did content analysis. So we do content analysis. We took DNA samples in order to understand the community there and to compare it with other sites. They have collaboration with the Patras University, with Padova University in Italy. They're funding, they have fundings from Shark Fund, Ocean Care, and yes, so they do also these studies with, for example, with Padova University. They also have this species. So they do these collaborations with other sites in order to also understand what I was mentioning about the DNA, so about the populations and yes, also the interactions with them. They also do a tagging program. So they tag the sharks with like the how do you call it? Yeah, like just like the small tag for cap and recapture and tag and recapture. And they have a special site that they share with the Padova University where you have all of this information about this. So it's a very big organization that has a lot of collaborations around not only Greece but also around Europe, and it's expanding as well.
Okay, good to know. I had another question. It's maybe more technical, but do you think that, so we speak about the spatial distribution, do you think that over time and links to climate change and so on, the distribution will change and maybe will affect it? How is your point of view about that?
Yeah, I think climate change will affect it because this is a very special goal. It actually 50% of the waters are impacted by hypoxic conditions, and so and around 30% of them, or a bit less, with anoxic conditions, because of past history of anthropogenic causes of release of nutrients, waste problems in waste management, and also aquaculture in the Gulf. So it already has a lot of problems. And yesterday, honestly, I was actually thinking about this. And I think also some papers were mentioning that seasonally already the hypoxic levels in the Gulf, they raise. So this will really impact the individuals, mainly if we start having climate change. And so the hypoxic levels may even increase, making the distribution of these sharks even smaller. And just to take into account, they say that just in the first 0 to 20 meters, they are like the good conditions. So you have very productive waters only until the first 20 meters. Until below the 20 meters, you start having hypoxic and then anoxic conditions. So for sure, climate change will impact the distribution of these individuals.
Okay, yeah, sure. Thank you, Andrea. So if I sum up, the species of the shark that you study, it is endangered, right? What is the conservation point? Sorry.
Can you repeat, please? I didn't hear that.
Sorry. On the first slide, we just talk about the conservation status of this shark.
It's endangered. It's endangered globally, but it's endangered globally, but in the Mediterranean, it's vulnerable. However, with the studies done by IC and in the area, it's also endangered locally in Greece.
Okay, so it is in danger, and fishermen, they can fish it anyway. So I understand that it's quite tricky, and yes, the regulation, yes, it's hard to follow in this case.
Yeah.
Thank you, Juliette. Thank you. I heard from my neighbor that my neighbor does have another question before I give back the floor to the audience.
You kind of just answered it though with because my question was I was looking at the distribution here. It goes all the way down to South Africa. So I was wondering about the kind of regional vulnerability of it. Is there places where it's not vulnerable?
Yeah, yes, as I mentioned, even in the IUCN, you can check like globally it's endangered, but in the Mediterranean, it's said to be only vulnerable. But
and is there any other information about similar to what you've done, but in other locations in South Africa and the north of Africa, on nursery grounds? Is there any information available you can compare to?
I would say like the main information was more about not about like more like the vulnerability of them, but more like their presence in these other sites, but more like presence, substance, like they saw them in all of these sites. But yeah, I couldn't conclude anything.
Next time.
Yeah, for sure.
So you still have a couple of questions. We're not going in a break with questions from the audience.
So Stephen mentioned this a little bit, but you mentioned that the species are declining over time, and I was curious if the fishing of them had changed over time, or if the consumption had changed in the population.
Yeah, well, in terms of fishing, I didn't present it, but in terms of the catch per unit effort, I had significant differences between years, between 2022 and 2023, and 2023 and 2025. And indeed, it has been changing a lot. And also like the sizes of the sharks. And yes, I would say the meat consumption is not that high. Estilodicta material, like you see in proportion of the 1,247 fishing trips, is very minimal. It's very minimal, the proportion of these targeted catch. But I would say what I wanted to highlight is the fact that they're also catching the pregnant females with the pups. So it's not as an intense fishery for eating the meat, but it's well known that in Greece they consume shark, so-called galeos. They call it galeos, and they eat it. It's like a cultural thing. Yeah.
Corina.
Really good work. I think this will be popular. And you mentioned they only do the basic tags for catch and release, but you think it would? I think I understood. You think it'd be worth to be able to track their movement, and if they use it as a general habitat, if they come back, if they go outside, etc. Do you think it's feasible as well for them? Do they have it planned? I know it's a bit more costly.
Yeah, mainly that's the main problem. And yes, we have talked about it with Martina, and we say that it would be amazing. to track them, to really see their distribution if they're going out. But I think a very good first step is now what they're doing with the DNA and seeing that, for example, that knowing that this population is highly isolated from populations outside, already without knowing if they live or stay in the Gulf, it already highlights the importance of conserving this population. You know, however, I would say that it's important to understand maybe if they're interacting with other fisheries outside, when they're becoming like from juveniles to mature adults, are they going out and then are they interacting with other fisheries or are they facing other pressures? I think it will be very good. However, I would say like also in the Gulf, they have 9 other rare species. So Mustelus is not the main, it's not the only species that is being considered under this project. So, but I think tagging is one of the things that with Martina we have talked and we would love.
Yeah, would be the cherry on the cake.
Yes. And maybe also we have a bit bigger individuals as well, because if you see they're very small individuals, so we would require the very small satellite tags that maybe they're way more expensive.
Okay, very last question. Time for last question. Yes, oh my God.
Should the fishing of this species be banned in the area? Have you talked already to the fishermen, or do you have ideas of what they could turn to? Those ones that are targeting the species?
That's a very good question, and it's something that yesterday I was also talking with Martina about. And indeed, only around 10 to 15% of the fishers in the area do it as their main income, as their main source of income. And the rest, they do it either as a secondary activity or as a secondary income. So honestly, the fishing activities is starting to be lost in the area. So also, I feel it's very like drastic for them to tell them like you cannot fish them. Also, because as you can see, the majority of them is bycatch. It's not targeted catch. So what also I see they have been doing is to help them, and I was actually part of one of the workshops to improve the handling practices to release the sharks. So when they get them in the net, they're teaching them how to release them. They even came to France to show fishers here how to release sharks and rays, because we have seen that this helps a lot in their survival. If you do it instantly, as the fishing time and the time the nets are in the water is not that much. When I was there, they left them maybe for 1 hour, 2 hours. So when they come back, if you release them immediately, the impact of the fishing activity is gonna be less rather than waiting a long time and then seeing if you release them. But if you improve the handling practices, I think that is like kind of like the first step. And then I would say first ban the direct catch, because bycatch is very difficult. And then what I mentioned, apply more bycatch reduction technologies. Yeah.
Okay, I think we're good. We have no question for you.
Thank
Now,
you.
congratulations, Andrea. We're now going to take a break. 15 minutes, we're coming back here. So also this message is for Stina, Juliette, Dominique, and people online at 10:15. 10:15. Okay. So we'll be back in a few minutes. I think we're good. So Jacob, you're back for your presentation, 15 minutes. Here we go.
All right. Hello and welcome to this penultimate session of today and this year of Immersion projects, indeed. I'm Jakob Röcke, and I'd like to present you my work on the predation effects that crabs have on hawksbill turtle nesting success in the Seychelles on a small place called Frigate Island. But let's start right into it and have a look at the background and why we talk about hawksbill turtles today. The main reason is because they are really important for reef health. And why so? Because they feed on sponges. Because otherwise these sponges tend to overgrow corals and thereby reduce these crucial habitats that are a host of such large portions of the biodiversity on our planet. Now, they also are critically endangered, and living on a small island like Frigate, the question we asked ourselves is quite simple. How are they doing? Because they come to Frigate to nest every year, and quite numerously so. The usual approach would be to just count them. But in ecology, we have a problem here because they migrate, and they migrate very far, in fact, thousands of kilometers sometimes, and that makes the usual approach impossible. So we needed to find other methods to answer the question of how the population of hawksbill turtles around Frigate Island is doing. And that's why we decided to identify and count the nesting individuals that come on shore during the nesting season, and why we excavate nests at the end of the incubation period to see how the nest has been doing and to assess the nesting success. Because, as you may all be aware of, reproduction, especially for such a species like hawksbill turtles, is crucial when it comes to population health. Now, that leads us to the next point, the threats, because of course, not all the eggs turn into adult and sexually mature individuals. So where can things go wrong? First of all, unfortunately, the top of the list is made by us, it's humans. We have been eating them for quite a while, and we've hunted them for their shell as well. In fact, the very glasses you might wear at the moment might include tortoise shell, how it's called. Now, these days it's plastic, but it used to be made not from tortoises, but from the carapace of hawksbill sea turtles. Another thing is that we need to consider is poaching of eggs. We've heard it yesterday from Corinna. It's quite frequent because they are very tasty, apparently, and therefore interesting commercially. However, we're on a small private island, so the risk of people actually poaching the nests is very low. So that leaves us with the environment as a threat, of course, because not only humans can be cruel, nature as well. Beach erosion is a major, major threat when it comes to the nests on the beach. Why is this? You can see this beautiful picture on the left. There used to be a meter of sand that just 2 months ago, when the hawksbill turtles came to nest, and now it's only bare rocks. So the currents really have a major impact as well when it comes to the nest site location. But there's little we can do except for relocation, and this is something I want you to keep in mind for later. But that leads us to the last main category of threats, and it's animals. Mammals, like rats, are known to predate on hawksbill turtle nests. And this is where the position of Frigate Island comes in, because we're on this little small dot in the middle of the Indian Ocean, about 50 kilometers away from Mahe, the main island of Seychelles. And so you might think, okay, we're safe, right? It's far away from everything. Yes and no, because of course, on a small island where you have people, we'll need contact to the outer world. So there are on a daily basis, there are boats coming in, bringing cargo, bringing food, bringing construction material, whatever it is. So rats are a serious threat because they can travel easily on boats. But luckily, we have extensive biocontrol mechanisms on the island that allow for a decontamination zone right around the port, where every box of bananas, every box of flowers, everything will be scrutinized for traces of other invasive species like rats or even plants. So that leaves us with one main threat from the animal section, and it's crabs, in fact. They are non-invasive, but they might be quite significant when it comes to the nesting success. So the question is quite simply, how do we study crab effects? Let's start with a simple assumption. More crabs will need more food. So now we need to find out how many crabs there are to have an idea whether there's a correlation between the crabs and the nesting success in hawksbill sea turtles. And we're in ecology again, so let's count them. But there is a problem. Everybody tends to count slightly differently. You might think from 1 to 10, it's fairly okay, we can manage, but it turns out it's not that easy. So what we needed was a very clear protocol to allow for the minimized differences in counting throughout the different survey events. And that's where the protocol comes in. I'd like to show you just a schematic of it. This is the main nesting site for the hawksbill sea turtle on the island. And we divided the beach into 4 zones, and each of which was then divided as well by the high tide line, being this part submerged by water during high tide, and this part usually being dry. This is the area where we see all the nests of hawksbill sea turtles. The crabs tend to populate both, but will come to the high tide part during high sea. Now, having these 8 subzones, let's say, we came there once a month to count the individuals on a low tide event in the evening. We threw an object into each of the zones to mark the center of a 5 by 5 meter square, in which we then counted the individuals for 10 minutes after a 5-minute acclimation period. Using the formula on the top right, we were then able to calculate the total number of crabs on the beach, and we did so by first measuring each of the zones with GPS and then calculating the surface area with QGIS. Now, knowing that, let's jump back to a bit of context regarding the turtles. My supervisor, Anna Zora, has been on the island for quite some time, and during the 5 years she monitored turtle nests with the conservation team on the island, we have seen a steep increase in the number of nests monitored on grounds. At the same time, though, we have also seen a drastic decrease in the average size of these nests. And when I speak of nest size, I mean the unhatched eggs that we find at the end of the excavation and the individuals that we extract. So given these numbers, the question is, what is causing this decline? And I've already spoiled it perhaps. One suspect is the ghost crab, in fact. Now, let's take a closer look. During the months that preceded my internship, so in the last winter or the summer of the Seychelles, we saw fluctuation in the average nest size. And at the same time, we saw also a fluctuation in the average number of crabs on the beach. And you might, okay, cool. Is he done? Not quite, because is it the full truth? Let's look into more detail. This is what I just showed you. And we didn't stop monitoring the crab when I arrived. That would be a bit silly because that was my project. So we continued throughout the months of April to July, of course, as well. And here's what we found. We saw a steep peak following the months of December, January, February, and March. And this is quite interesting because those months here would be considered the peak season of nesting behavior of the hawksbill sea turtle. So, and this on the other side, the off season. So the question is, why would we see very little crabs at the time where there are loads of turtles and turtle eggs around, and so many more crabs when they are actually all gone already? That leads us to a first interpretation of this background information, and it's first, of course, would be crabs have actually no effect on the nesting success, but it seems a bit unlikely as they are the only net predator that we have on the island. So that brings us to possibility 2, which is that crabs might actually be where the food is. Now, this is just a hypothesis, but maybe they are underground during the peak season when all the nests are full of turtle eggs and plenty of food, basically, underground. And they come up overground once those food sources are depleted. Because, as I mentioned earlier, for the protocol, we counted the individuals on a 5 by 5 meter square, not digging into the sand. We just counted on the surface the crabs that we saw. However, it's difficult to assess how many are, in fact, underground. That leads us to what we can do to improve the situation or close this gap. And it's quite simply to include holes on barrows that are within these plots into the survey event, so we can infer from that point onto how many crabs might be hiding underneath the sand currently predating our nests. And because we don't want to excavate a 5 by 5 meter plot every time we do a survey. We also wanted, but still want to validate whether there are crabs underground. We will also include the crab count during the excavation of the turtle nests. So whenever we excavate a turtle nest at the end of the period, we will now systematically record how many crabs we found in the nest, or whether at all, and how many burrows went down to that nest chamber. This is data that has not yet been structurally recorded, and this is something you might want to keep in mind for later. Now, being a conservation island, Frigate and the team on Frigate around Anasora, my supervisor, wondered, okay, what can we do? Because after all, it's a critically endangered species, as I mentioned in the beginning. And the starting point, or the first word that comes to mind for people who have heard about turtles, is hatchery. So a protected area where you translocate nests into to make sure that they are exposed to lower threats. Now, on Cousine Island, which isn't too far, I showed it on the map as well a moment ago, it showed that it could decrease the crab influence. And on Frigate, we already established a hatchery in 2022. So the question was quite simple: have these translocations since then had a positive effect, or could they reduce the crab effects on these nests on the island? And to check out whether that is actually the case, the task is simple. We need to compare the available 3 seasons of data from the hatchery with the wild. However, there's a slight problem, because what do we usually compare? It's called hatching success, and it's usually defined as the living individuals leaving a nest. But it's a small island and we have limited resources, so we're not always there when the hatchlings happen. So the exact number of hatchlings are unknown. And now people who have worked with turtles before might say, but you can count the eggshells. That's true, and that's what we do as well. However, when you see an eggshell that is broken because it was hatched, and you see an eggshell that is broken because it was predated by a crab, it is possible to tell the difference, but it's very tricky. And that leads to the fact that the actual number of live hatchlings is very hard to report. So we needed to find proxies to actually tell what the hatching success was. Now that leads me to the predation indicators. One that I'd like to present is the presence of crabs or holes in the nest. I already mentioned it. It's something we want to record in the future, and that we have recorded so far only incidentally, so in the comments section. And from the comments that I read through, I defined 2 cases that we could have. It's one, yes, there were crab holes in the nest, or no, there were no crabs or holes in the nest. And looking at the nest size again, that we already had a look at earlier, we can see that nests with no crabs or holes show a significantly higher median than nests with no crabs or holes. So that's already an indicator that, yes, we're onto something. That leads me to the second predation indicator that I'd like to introduce to you, which is the predation score. It's something we assign to every nest at the excavation, and it ranges from 0 to 5, with 0 being no predation at all, and 5, everything has been predated, nothing left, and the 4 intermediate levels for each quartile of the range. Now, if we do the same comparison for the nest size for these 6 categories, we find a cascade effect that the higher the predation score, the lower the median nest size. Now, of course, you're scientists, so you'll notice the letters on top of the box plots, and you'll see that indeed the predation levels 0 and 1 are not statistically speaking significantly different. So is 2 and 3 from each other. So technically we have 4 main groups of predation. However, because 0 means no signs of predation whatsoever, I decided to treat it differently for the further analysis anyway. Leading me to the final graph I want to show to you today, which is about the comparison of the hatchery and the wild. And you can see here the nests, the portion of nests that fall into each of the just presented 5 categories, which is the main result, the main takeaway is that all the wild nests show a much higher portion of completely predated nests in comparison to the hatchery throughout the whole years. Same is accounts for the not predated at all portion of both, where the hatchery always outcompetes the wild. However, it should be mentioned that the fluctuation is quite significant, and I'd be very open to discuss possible factors in the question section. But the key takeaway for Frigate as a conservation island is that the efforts in protecting the endangered hawksbill sea turtle are in fact working. So that's good news. And that brings me just to the last takeaway and the summary of it. So during the last 5 years, we've seen an increase in the nest numbers, but a decrease in the nest size. In the last 3 years, the hatchery nest showed less predation than the wild nests. And just over the last year, we could see that the crab abundance fluctuated in relation to nest size with no definite resolution with the data available so far. Going forward from this, we will augment the protocols that I mentioned already, and this will allow us to make more conclusive, more conclusive in whatever analysis. Thank you so much. At this point, sorry. So this is the moment I'd like to thank Anna Zora, my supervisor on Frigate Island, who guided me through this whole process and was there for any questions that I had. I would also like the present, past, and future conservation interns on the island, as without them, this data would not be there. And of course, sorry, there we go. And of course, the Frigate Island Sanctuary, which hosted me on the island for 5 months. And of course, Christelle, Christophe, Thomas, the coordination team in general, and Université Côte d'Azur, and you for listening. Thank you so much.
Okay,
Okay.
thank you. So we can start with Dominique and jump into USs.
Okay, thank you. Very interesting study and the link, and apparently also the complexity of trying to see whether there is a link with crab population and behavior and the nests. I started to write down a couple of questions, but then you answered them, and then new questions popped up. Well, maybe I can start with one of the last slide in the conclusions. You indicate that, so there has been an increase in nest size, but with, no, what? Can you show the slide? Yeah. There was an increase in
nest numbers.
Nest numbers and a decrease in nest size.
Yes.
Could that be an indication of a behavioral response by the turtles in minimizing predation risks by making smaller nest sizes, because maybe they're less easily to be detected? And yeah, I don't know if like, for example, it has already been observed that instead of making one big nest by a single turtle, that they make 2 small nests.
Yeah, it's a very interesting point you're raising, and it's something we thought about a lot as well, because a nesting event often takes many, many hours and is very, very energy intensive and resource intensive for a turtle to undergo. Now, usually in one nesting season, a turtle would come up to around 3 times to the same beach to lay nests. So you're right that there are several events. However, just from the data that I analyzed, I couldn't tell an increase in the nesting events of single individuals throughout those 5 years, though this is something that could be looked into with more detail. I also, looking for an answer to this, went through some literature and found that the carapace size has seemed to decrease and has been proved to be correlated to the batch size of the nests. So there is literature suggesting that the population around the Seychelles might currently be either younger, so smaller therefore, or just smaller in general than previous generations of nesting turtles, and thereby decreasing the nest size.
Okay.
But, it would be very interesting to look at it from the angle you just described.
Yeah, and do you know anything about egg size? Because if you have different reproductive strategies of less eggs, but maybe then with more yolk, so more energy, more chances of survival, RK strategies, whether this could also be involved or not?
It's nothing I looked into, but it's a very interesting point, and I think for future studies, potentially realizable, though difficult, because we excavate, I think in the last season it was around 300 nests or so, and that's quite some work during just 4 months, and it would involve a proper analysis of the remaining eggs. And I think if we could manage or make available the funds, it would be a very interesting project to pursue. Though I can't tell you exactly how it turns out.
Maybe a last one. In one of the last graphs where you showed the different predation levels over 3 years, if I understood correctly, and there was this quite some fluctuation in the number, this one, the number of nests where there's total predation. Do you have any idea whether this yearly fluctuation also reflects yearly fluctuation in crab population densities?
Unfortunately, we did not have the crab protocol in place at that point, but there's a reason why we started the crab project in the first place, which is that this year showed so much more predation than the previous one. And one aspect why the level of predation in the hatchery is so much higher, one hypothesis that we came up with is that we in fact had the hatchery in that year in the same location as this one. So there might be a chance that the crabs learned about the fact that this area of the beach is particularly worth it when it comes to burrowing. However, this is just 2 years, so it's hard to make a statistical analysis on it. But we also see a much higher predation in the wild as well. So the factors might be also found in the threats that I mentioned in the beginning and the other threats. So be it temperature, humidity. I discussed it quite a bit with my supervisor as well, how the weather conditions changed that year or not. However, even though I had the ambition of doing a multivariate analysis in the beginning, it quickly proved to be hardly feasible with the level of data that I eventually extracted.
Okay, thank you.
So Aziz, and after Aziz, we'll go to Julien.
Super cool. I like it. I have a question about the relationship between the increase in number of nests and then the decrease in size. When this data was collected from one location?
Yes.
The same location?
Yes.
So how big is the beach?
The beach, wait, I'll show you. Is this one. Okay, so this now you see on the photo, you see about half the beach. And here you see the whole extent. This is the size of it. The number, I don't have it on top of my head of square meters.
But it's density dependent.
Yes.
Yeah. Do you think that plays a big role in it? If we have a number of increase in nests, then the size of the nests will have to be possible as well. I mean, you have 187 nests on this beach.
Yes. So there's a possibility that, and that's something we try to avoid as well. When turtles come on shore, they look for the best spot to lay their nest, of course. However, and so that means they sometimes check out one, they crawl further, they check out another, and then they decide to nest somewhere. And the further the season progresses, the higher the probability that one turtle will lay its nest onto another one, and thereby essentially excavate and destroy the first nest. So it could definitely lead to the fact that nests or eggs would be lost already during that process.
It could be just a simple density dependent. Yeah, possibly. Yeah.
Yeah, no, absolutely.
And for the turtles themselves, are there any residents that stay around the island? They don't leave. Yeah.
As a matter of fact, I spotted quite one individual definitely repeatedly just swimming across the reef offshore, which is quite lovely.
Is there any facial ID?
We don't have facial ID, but we do have tagging. So whenever a turtle comes on shore, There are protocols in collaboration with the Turtle Action Group Seychelles, co-managed by my supervisor and Jean Mortimer, that make sure that these turtles, if they show up elsewhere, are possibly recognized. And data on migration can be collected.
So you had two possibilities there for the crabs.
Yeah.
I'm going to ask a really mean question.
Go ahead.
Where are the seabirds?
Funnily enough, they're not part of the picture. There are quite large numbers of seabirds, but we have never seen them predating on the hatchlings when they fall to the sea, which is impressive because it's a major threat elsewhere, but not on frigate.
You have rats on the island?
No.
There's no rats.
There's no rats. There are no mammalian predators whatsoever. All the cats that were once there are gone. All the rats have been eradicated after a short invasion in the late 90s.
Yeah.
And ever since, the protocol has enabled the island to stay rat-free, which is quite important for...
Okay.
Yeah.
So possible that the seabirds don't have any influence on the...
That's how it looks. It also would affect mostly the... how can I say? The later part of the hatching success, so when they crawl to the sea. The nesting success itself wouldn't be affected because they usually don't dig, the most birds at least.
Yeah. Okay, because it's very different from the Chagos. The Chagos is quite famous for rats, seabirds.
Exactly.
Yeah. My final question is on the recruitment timing of the crabs.
Yes.
When is the recruitment?
I looked into that, and it's in fact too short to be just about, wait, it's the graph. Whether it's whether we could see essentially recruitment here or like reproduction here and see the recruitment here, and it doesn't match. So
when is the recruitment time?
I don't have it at the top of my head, but I can come back to you with the answer.
Okay.
Yeah.
And is there anything predating on the crabs?
Sorry?
Anything predating on the crab?
Not that I know of on the island, no.
It's possible that the crabs are recruiting the whole year.
Yes, it is.
So the dynamics is determined by, maybe it's not recruitment.
Yes.
It's maybe you're onto something. But.
Yeah, because we would assume that we would see a lot smaller crabs here then, if those were the recruitment's kicking in. Though this hasn't been a specific data point collected, just from the observations of being there, this is nothing that I noticed. Yeah.
Okay. Thank you very much.
So Juliette, finally Stina.
Okay, Jacob, thank you very much for this nice presentation. I have just a few comments about just the presentation in itself. So maybe your PowerPoint could include more information, like the slide number was not easy to find, and moreover when we had picture, sorry for that. Maybe you can also have title for the, like Mat and Med or introduction and so on. Not so hard, not so easy to follow in this case, but for me, it's just my tips for you as you are in Master 2 now. So linking to the question, same question that before, I don't really understand the entity and the organization about how did you make this job, why it is entering into a huge program or no. Can you explain a bit about that, please?
Yes, of course. Where do I start? So we find ourselves on this little island in the middle of the Indian Ocean, which happens to be, well, I don't know what it is anyway, which happens to be privately owned. So I was part of the conservation team on site, and this team is fulfilling the function of trying to maintain the best ecological state of the island, whilst also enabling the use of the island for a hotel. And within this framework, we worked together on the various aspects of the island life. So we also looked a lot into the endemic bird species that we have, into endemic snail species, into all parts of the ecosystem, and the turtles are one aspect of it. And to be able to report the state of the ecosystem, this is how the turtle monitoring program comes in.
Okay, so
Um,
it's funding by the hotel, if I sum up.
sorry?
It is funding by the hotel.
You could say so, yes.
Yeah. Okay, let's say it in this case.
Yeah.
I'm very amazed about this like crab problem. I'm sure you already developed it, and I'm sorry it was the case, but maybe you can let me know a bit about. Okay, so we have the crab. There is no predator. Human can be a predator, yes or no? Maybe they can have trap. So, what is your point of view about that?
On this island, as it is private and has its own security team, usually there are no predator, like no humans that come and interfere with the nests. Of course, you may have guests of the hotel, but they're rather not known to be digging in the sand for the nests. And would also be halted in their actions by the staff. But yes, of course, the factors that I mentioned are quite, quite drastic in other places. And I know of people living on the island who are Seychellois that, yes, even though it's illegal in Seychelles, the poaching of eggs and the trade of hawksbill sea turtles, that usually they somehow know how it tastes.
Juliette was speaking about the crab.
Sorry,
yes,
predate on the crab.
I'm sorry. Could you say it again?
Sure. So about the crab problematic, you think that firstly crab can be eaten by local? Well, you told me that local are not eating crab.
Sorry. That's
And
a
also
misunderstanding.
maybe some crab trap or stuff like that.
Now, okay, sorry, I was thinking about the turtles. My apologies. The ghost crabs are not eaten by locals. There's another crab type that is, but it's not known to predate on the nests. In terms of traps, it is something, of course, we have thought of. However, as they are an endemic species and a natural part of the environment, it feels too invasive to go to these extensive measures. Albeit it might be nice to strengthen the population of hawksbill sea turtles, this is a level of interference that, would definitely ensue further ethical considerations that, we're not okay with so far.
Thank you very much, Jacob.
Thank you, Juliette. Stina, the floor is yours.
Hi, Jakob. Thank you for this really nice presentation. Dominique stole my maternal investment questions. So, but I was thinking, because it kind of looks like a trade-off, right? In nest size and nest quantity. And you said you're not really doing any work on the eggs itself to have a look in, like, if there's differences among different females or if there's multiple nests from the same female.
No, not so far. However, how can I? No, we do tag the individuals when they come on shore. However, we do have also untagged individuals, so we don't know whether they've been there the year before. But it would be a very interesting angle to look at it. Another aspect that we recorded instead was the developmental stage, because what we found was quite varying as well is the actual emergence success. So not only just the nest size, because now I looked at nest size because I really wanted to isolate predation as a factor. But what seems to be more dominant, just from observations, is unfertilized eggs in comparison to fertilized eggs between the nests. Yeah.
Cool. So you're tagging the females. Are you also doing any type of measurements on them, or is this
Yes,
really
It goes along with. Let me check whether I have the forms and the annex. I should do, because I came towards the end of the season. I did not see any nesting turtles, but only excavated nests. So, but we have this protocol that allows us to record exactly where exactly we found it, what species of turtle, because we do have occasional green turtles coming by, as well as whether they only dug one pit or several. And And we do take the measurements of, where do we have it? Curved carapace length, curved carapace width. We record whether there are any injuries, as well as existing tags. Yeah, so that would be a separate, separate
That's
analysis.
very interesting because you said you're also doing some relocation of the nests.
Yes.
So you're handling the eggs already as well. So you technically could also do some measurement on eggs. And I don't know about turtle eggs at all, but maybe you could like shine a light through and measure the diameter of the yolk or something to get like an idea of quality of the egg with yolk yolk diameter, maybe.
Yes, I think if I were to go through another turtle season, I think that would be a wonderful opportunity. I know from my colleagues who have been there through the peak season that it is utterly intense work already with the workload now. So I think it may come down to resource constraints eventually, but I think as a perspective, it's very interesting. Yeah.
Yeah. Do you know if the relocation of the eggs has any impact or harm on them? Like, is there a difference in hatch rate, or do you see anything?
There is. Well, I also try to look at that. However, to determine the hatch rate, I quickly touched upon it earlier. You need to compare it to the wild, and in the wild, we would not necessarily be there when they hatch. So the eggshells that we find could either be from hatched, like after a hatching event, or after a predation event. The learned eye will be able to tell the difference, but as we have a rolling basis of interns coming through the years, I found the data to be difficult to interpret on that exact parameter to make sure it's actually reliable. So I excluded it.
Okay. And then one very last question, and you might have said it and I might have missed it, and then I'm sorry, because then this will be a stupid question. But is there any, because you say to measure like crab abundance, you look for crab holes in the nests, right? But is there any like observed predations of the crabs on the eggs or hatchlings? Or
yes, we can. Yeah.
What's the sign that the crabs are actually predating on them?
You can observe it at different stages. Let's say the first and most noticeable event would be when the hatchlings crawl to the sea. You would see ghost crabs rushing there to grab them, carry them away, and bite the head off. It's cruel to watch, but it's nature. So that's one option, and then of course when you excavate, you can see the burrows going down to the nest chamber, and you'll have them hiding. Of course, when you dig, it's a bit of a mess, so you don't exactly see what the crab has been doing, but you will find usually broken eggs, especially when there's a crab right inside, because it will not have eaten the entire egg by then. So that would be a clear indicator. Sorry, there was another aspect to your question, I think, but I forgot it.
No, I think you answered it. Thank you. That's it for me. Thank you.
So we have time for one or two questions from the audience. Andrea.
Thank you, Jacob, for the presentation. Now that you mentioned these crabs going behind the turtles, I'm wondering, how big are these crabs compared to the hatching turtles?
Yeah, they are actually not that big. I don't have them measured in centimeters, but if you take this is the size of a normal ghost crab, and then you have the size of an egg, the size of a hatchling. So it's a decent size for a meal.
Yeah.
But there are many ghost crabs. So as they are scavengers, I assume that they eat as much as they can. They leave it, another crab comes. And we also saw in the beginning, you might have noticed in the introduction, there was a picture of hermit crabs. We don't see them burrowing down, but if they have the opportunity to eat an already dead hatchling, they will. So that we already also recorded their presence. But yeah.
And just like to follow, so mainly the predation will occur inland. Like as soon as they will get to the water, then they will.
That's another story.
Or they also like what is kind of like the story?
You're the shark expert.
In terms of like the crabs, like as soon as they reach the water, they're safe from the crabs.
They are safe from the crabs,
Exactly.
but that's usually the moment where you can see the first black-tip reef shark coming, and then another one, and then another one. So it's quite cruel.
I like that movie.
Yeah,
The real fun.
yeah, it's a bit of a pedal run. So yeah.
Okay, thank you. Last question, or no? So you're
That's-
ask.
Just a really tiny one, just because you said you saw these different levels of predation on the eggs and the. Do you have any pictures of that? And did you not show them because they were like too horrifying?
No, I didn't take any pictures of bitten off heads of hatchlings. And the nests themselves, it's quite a mess if you have all the sand in there. You would need to clean it first, and it's tricky. So no, I'm afraid I can't show you any graphic details.
We have to go, I think, and I guess we're maybe some also all eager to go for the last presentation. Thank you again, Jacob. So with no further ado, and surprise, Raphael, you have the big honor to conclude this 7 sessions of Immersion project.
Yes.
And with Raphael, we're coming back to Massachusetts, but from Massachusetts this time.
Hey, hello. Today I'll be presenting my immersion project where I investigated octopus sucker recruitment patterns across different regions of the arm in Woods Hole at the Marine Biological Laboratory. So first of all, what is sucker recruitment? So sucker recruitment is as simple as when an octopus will attach or use one of their sucker on a surface or an object. And why would one want to study sucker recruitment? Beyond just being fundamental behavioral research, it can also inform soft robotics. So soft robotics is the field where engineers make robots that have a soft structure, the same, inspired by octopus, right? And studying how specific suckers are recruited and patterns that they can have can inform engineers in making robots that behave or act the same way. Studying sucker recruitment can also inform potential neuroscience research because by studying behavior, you can uncover potential underlying pathways in neuroscience and in the nervous system of cephalopods. So how would one characterize sucker recruitment? So this being an octopus arm, here where the suckers are a bit bigger is towards the body, and where it gets smaller is towards the tip of the arm. In green is the focal sucker, so that's the first sucker to attach to a surface. The first thing you can characterize is the pattern. So the pattern would be the sequence in which these suckers will attach. Then you can also look at the type of attachment. So this can be ipsilateral attachment. So ipsilateral attachment in purple here is related to the previously attached sucker. It's a sucker that attaches on the same row of suckers. And you can also have contralateral attachment, which is when a sucker attaches on the opposing row from the previously attached sucker. And this can also be analyzed according to the direction of these sequences. So either towards the body or towards the arm tip. And then finally, you can also look at the latency. So the latency is the timing between each sucker attachment. So the time between two suckers attaching, and this can be characterized according to the site of attachment and direction, but it can also be characterized according to the arm region. So an octopus arm is divided in 3/3. The first one being the tip of the arm, the distal region, then you have the medial region, and then the one closest to the body, the proximal region. But for the purpose of my study, I have focused on the distal and medial regions. So my research question was to provide insight on sucker recruitment dynamics across the medial and distal region of the arms. To do this, I looked at the patterns of the sucker recruitment, so the sequences in which they attach. I looked at the type of attachment, so the frequency of ipsilateral versus contralateral recruitments, and I looked at the latency of time between sucker recruitments. So how did I do this? To do this, I did a whole life animal behavioral study using the species Octopus bimaculotus, and the aim was to have them do this wrapping motion around this disc, as you can see this. So what we did is for each trial, we made 8 agarose discs that we infused with shrimp extract, and we put a little piece of shrimp so that it would attract the octopus. And then we place them in the corner of the tank, 4 on each wall, and we covered the rest of the tank to minimize distraction. And we placed 2 cameras, one pointing each wall corner. And these cameras were filming at 60 frames per second. And then what we considered to be a good clip, so a full sequence that we would analyze, was when 5 suckers were recruited. So we had the first sucker, the focal sucker that would attach, and then we had 5 following suckers, 5 following recruitments that would attach, and that we would consider to be a good clip for analysis. And then we characterize these clips according to the arm region, so either distal, the tip of the arm, or the medial, the middle region, and according to the direction of recruitment, so towards the tip of the arm or towards the body. And then we analyzed these videos following a naming convention that I can get into later if you have any questions on it. So first of all, looking at the pattern. So the sequences in which these suckers are recruited. The most common among all regions, so distal and medial, and in both directions, was this strictly ipsilateral recruitment pattern. So where suckers are recruited only on the same row, 5 different recruitments on the same row of suckers. Now looking into the distal region in the direction towards the arm tip, these 2 other patterns were seen frequently. As you can see, there's a lot of ipsilateral same row recruitments here. In the same arm region, but towards the body in the other direction, this was another sequence that was frequent. As you can see, first attachment is also ipsilateral. Then going into the medial region, but towards the arm tip, this was another pattern that was frequent, a lot of ipsilateral attachments. So these are a lot of patterns to look at and take in at once. So what I want to highlight here is the one that's common across all our results and categories of direction and arm region is this strictly ipsilateral attachment. And among the other regions, the other patterns that are also frequent, the first recruitment was always ipsilateral. So this might suggest a potential narrow pathway between these neighboring ipsilateral suckers. Now looking into, so I mentioned we had a sequence of 5 suckers, right? So when we had a sequence of 5 suckers, what was the frequency of the recruitment being ipsilateral, so same row, or contralateral, cross row recruitment? And in the distal region of the arm, in both directions, the ipsilateral attachments, so same row recruitments, were more frequent than the cross row recruitments. This was also the case for the medial region in the direction towards the arm tip, but the direction towards the body was not significant. This might just be due to a lower sample size due to our experimental design. So take away from this, similarly as we saw with the patterns, there seemed to be a tendency with these ipsilateral attachments that are very frequent. Now looking at the latency, so the timing between these attachments. First, looking into the distal region, we can see that comparing the 2 directions. So if we look towards the arm tip versus towards the body, it seems that the speed of recruitment is faster towards the arm tip than towards the body. And then if we compare the ipsilateral in purple to the contralateral, there seems to be a trend where the ipsilateral same row recruitments are faster than the contralateral. However, I do want to highlight these results here that came out significantly different, but when you visually look at them, they wouldn't appear to be, there wouldn't appear to be a visual difference between them. This might actually be a case of a false positive due to the correction method I used. I can talk a bit about that later as to why I decided to still stick with this correction method, but I do think there should be some caution in interpreting these results. Next, looking into the medial region. So just comparing the distal with the medial, you can see that the latency time of the medial region seems to be on average a bit higher than this distal region. And then if we look within the medial region, there are different trends than in the distal region, where the ipsilateral recruitments are faster towards the body. Now, we saw a lot of ipsilateral recruitment. So there were a lot of recruitments that were on the same row. And as I hinted to before, this might actually suggest a potential neural pathway between these ipsilateral suckers. When we look in literature, contralateral contralateral neural pathways have actually been described. but there is no existing description in literature yet for ipsilateral neural connections. This might just be due to the finer scale of these connections, and the method that was used in the paper that described these cross sucker connections use electron microscopy, which might not provide fine scale enough of results. So this kind of goes to show the multi-layer of organization that goes into the nervous system of an octopus, and how certain pathways might actually be on a finer scale or on different scales. But from a behavioral perspective, it would make a lot of sense that these neighboring suckers would have a direct neural pathway to link them, because it would facilitate the recruitment that we saw so commonly in our results, this ipsilateral same row recruitment. And now looking at the latency, so the speed in attachment. Our results showed that in the distal region, so in the region of the tip of the arm, the attachment latency was faster towards the arm tip. And when we look in literature, anything related to recruitment speed, or maybe speed in the arm of signals being transmitted, we find this study by Zugo and Al that shows that sensory signals are actually found to travel faster towards the body. So when I compare these, it might look contradicting, but I do want to highlight the fact that we looked at attachment frequency. So this is attachment latency. So this is the speed at which two suckers will attach. It doesn't necessarily highlight signal speed, which is what Letizia Zullo focused on in her study here. So it's interesting to compare both, because the attachment latency might reflect a signal being transmitted. But in this case, we're maybe hypothesizing that maybe this might not be the case. So what do the results actually mean? So the results that we had might actually reflect more of a motor coordination rather than a sensory coordination. So rather than a sensory signal being transmitted. And we saw these results in the distal region, and the distal region of an octopus's arm plays a key role in motor action and in the fetching motion. Which would then, when we think of our experimental design, the way they had to wrap their arm around a disc, it maybe recruited more of a motor reaction than a sensory stimulating input to them. So we're analyzing more of the motor coordination here. And then if we compared it with the distal and the medial speed of attachment, we saw that they had different results. And we can hypothesize that maybe different arm regions have different functions. And this is not based on literature, but just a hypothesis based on these observations, that maybe the distal region is used more for exploration or object manipulation, whereas the medial region might be more for processing or transferring information centrally. Yet again, this is not proven, it's just a theory. But these differences between these two arm regions in the speed might also be due to anatomical factors. So each sucker has these little neuronal segments, and each sucker has the same amount, same number of segments. So the smaller the suckers get towards the arm tip, the smaller these segments get. So the neural signals might actually travel faster in the distal region than in the medial region, where the suckers are a bit bigger. So there are a lot of limitations to this study, and I think that the main one would just be the nature of what we're studying is incredibly challenging, and having an experimental design where we get to have a very clear view on the suckers, and especially both row of suckers, to be able to analyze them and quantify them is really challenging. An octopus has many arms and is very flexible, and often will hide the suckers with its body. So we ended up deciding on this experimental design, which maybe led to more unequal sample sizes, or also favored distal recruitment, because we had a very limited number of samples in the medial region, which maybe decreased our significance in statistics. And this might be because the agarose discs we use are smaller and a bit closer together compared to medial suckers that are a bit bigger and might need more space to fully recruit 5 suckers. And it would get a bit crowded where a lot of arms would start interacting with them, where you wouldn't be able to get that full sequence of 5 attachments. So how can we improve this? Obviously, we could keep the same model, but improve the size of the discs, space them out a bit more, use high-speed cameras to have really precise quality of videos, or we could even come up with a new method where we could use a sensor-equipped discs to avoid the human error, and we wouldn't have to rely on what we see to be able to analyze these recruitments. And we could purely use the sensor that would give us information on what we're seeing. And there are many other ways that we brainstormed before coming up with this protocol, but there is a reason why, maybe due to limited resources, we ended up with this experimental design. So overall, we found our results point to a potential neural pathway that can link these ipsilateral neighboring suckers. And like I mentioned at the start, these behavioral studies can inspire potential future neuroscience studies. So this could maybe inspire studies where they would look on a finer scale to potentially find these pathways. And then having more information on the recruitment patterns and the ones that are more frequently used in this kind of wrapping motion in the octopus arm can inspire soft robotics to make precise robots that are used for this kind of movement. And overall, this is kind of one of the first studies looking at sucker recruitment in this specific design. So this study can provide iteration as for future studies to maybe improve or work on another similar study. So I'd like to thank my supervisors and thank Kendra, Ashley, Max, and Roger Hanlon, as well as the Maris team and all my classmates for making this happen. So thank you so much.
Thank you. So I propose we start with you, Juliette, for this last one.
Thank you very much, Raphael, for your presentation. It was very interesting to dive into this circular part, if I may say, of Octopus. My first question is the same that I asked to everybody now. So could you make contextualization of your project? Why it is so, firstly, the organization who make it, and why it is so important to make this kind of research? And yes.
Yeah. So I did this research at the Marine Biological Laboratory in Woods Hole, which is a private research lab. And this was part of, I think, a 2 or 3 year grant where they looked specifically at sucker recruitment. So exactly how the suckers are recruited. And like I mentioned at the start, it could be used for different usage, such as informing soft robotics. Yeah.
Very interesting. And so about can you go just in your Mat and Med? I was a bit curious about material and yes, please. Yes, sorry for going so many up, but can you just explain me a bit more about how was the experiment ongoing? And then, yes, just this part to just understand more about how it was going.
How I conducted the experiment?
Yes.
Or okay. Yeah, so what we did is we made these discs that we would do for every trial. So we used 5 different individual octopuses that we did 10 trials for each octopus. We would run like 3 octopuses per trial day, and they would do 1 trial per day. And essentially, we would put them in the experimental tank, and they were actually, this study works on a study that, well, it's a continuation, if you will, of a study that they already looked at sucker recruitment, but with a different experimental design where they had these octopuses do this wrapping motion. So the octopuses were already pretty trained. Some of them didn't want to cooperate, but when we would put them in the tank, they would go up to the discs pretty fast, which actually helped us in terms of like time management. But yeah, no, we would put them in the tank, and we would film on these 2 cameras at the 2 angles, and we would put a 15-minute timer because they would react right away, right? And then we would put them back in their home tank, and then these videos, we analyzed them frame by frame. So we exported them on Procreate, and actually I have like a picture at the end that we wrote on every image the frame number, as well as which we kind of like created a name for each sucker to be able to have a standardized result and grading criteria. This is kind of what I was referring to my method. And then we were 2 people grading these videos, and then we graded them independently. Then we came together, compared them, and came to an agreement, and then put them in the spreadsheet where it's a bit overwhelming to look at. But the key point was to highlight that the frames, we would be able to have that time and which sucker was attached. So then we could then create sequences and extract that. I don't know if that answers in the method.
Yes, thank you very much. It's very easier to understand with the Appendix 6. Thank you very much for this doc. So if I understand well, unfortunately, they have some like error, and this is quite understandable because a lot of image and so on. So to your point of view, what kind, like, how could you improve this work, and how could you maybe using EA or I don't know. So what is your point of view about that?
Yeah, so we did have some discussions of maybe using software that could recognize these suckers and could name them or grade the videos rather than us having to do it, to maybe have more of a standardized view. But the technology isn't there yet, and a lot of behavioral softwares such as Boris that Gaia mentioned can't do that yet. And we consider maybe AI, but then again, we didn't have like the knowledge to develop an AI, maybe, well, we didn't have it yet. Now we had AI classes, but yeah, no, yeah, so there are limitations regarding like how we can analyze a video. I do think to improve what we do have and what we did, I think it was just more time to have like a standardized amount of trials for each sequence, because that was kind of what was limiting, is that we had the same amount of trials, but not every octopus provided good clips that were usable, right? Because there was these 5 sucker recruitment clips that weren't always, it wasn't always the same. Like one day, maybe an octopus gave us 2 clips, another one gave us 5, maybe one just gave us 1. So I think it would just be doing the experiment on a longer period of time to really get more results and be able to have stronger stats afterwards.
Perfect. Thank you very much, Raphael. And well done for your PPT. You were excellent.
I say Dominique after? No, I don't remember. Dominique.
Okay, thank you. Very nice work, very interesting. Also because I'm particularly interested in biological grasping systems. So first thing I was wondering, so you could identify that the first sucker that was recruited, and then you always see that the next one is always on the ipsilateral side. I was wondering, do you have any idea about the longitudinal orientation of the arm if they attach? Do they do it flat and only attach with one sucker, or do they attach on a side, which then explains they start ipsilaterally?
Yeah, so I think this could be a limitation in itself of our study, is that we didn't distinguish these kind of approaches of recruitment. I can say that from the many clips I analyzed, it was a mix. It was a mix sometimes a very clear on top, very clean wrapping motion, but sometimes it was more like coming from the side, and you would see one sucker would like reach out and then it would wrap around. So I think maybe, obviously that would have an influence on, it would be interesting to look at then according to how they approach, what's the recruitment pattern afterwards, right? But unfortunately, we didn't do that for the study.
Yeah, because I was wondering, because we kind of see something similar in seahorses when they grasp. They do seem to do it with more on one side first and touch, and so that kind of suggests that there is some kind of sensory feedback that might help them if they can concentrate it more on a particular point. And that's bring me to the next question. At some point you said, based on the latency pattern that you see, you mentioned that it would be more about motor coordination rather than sensory. But do you have any idea to what degree there is a kind of immediate feedback loop of sensory perception and direct feedback on motor control, so that they kind of modulate individual suckers depending on how they are sensing at the sucker level? Do you have any idea?
Well, obviously, I concluded that my results were highlighting motor coordination, but I do obviously think that motor movements is highly correlated with the stimuli they're also getting, and everything's kind of happening at the same time. So it's hard to distinguish that. But I do think what we found I highlighted more as a motor coordination because we were looking more at this movement. And I think it might actually be the experimental design that favors this just wrapping motion towards the tip of the arm rather than wrapping the rest of the arm. You might just feel so because of it might be like easier to wrap this than wrap the other because the suckers are a bit bigger towards the body. But I do think there must be like a certain feedback loop of like, okay, like they had a first contact with this disc and there is like a stimuli, right? And they are able to be like, oh, like this kind of like has a certain texture or has a certain like, we put like prey extract into it. So I do think it's highly linked, which is why it's kind of hard to interpret these results because there's so much under, it's like touching the surface, right? That what we're seeing, I can like only deduce is like, okay, well, maybe this is what's happening and you might reflect more motor, but I do think it would be interesting to also look more at stimuli, but with this kind of behavioral design that we had, it's hard to also look at how the sensory is being transmitted in the case.
Okay. And then do I understand correctly that the recruitment latency that you quantified is the time difference between two consecutive suckers that were attached?
Yes.
Yeah,
yes.
So that time difference. Okay.
Yeah.
So how variable is this? Do you have any idea? Let's say if you at suckers at similar location among the arm where they would start, do you see a consistent latency for comparable suckers, and not comparing distal with medial, but if you would look at medial ones, is there a consistency latency time between them, or is this quite variable? Because the first could be, if it's consistent, it could indicate that it's a fixed motor pattern. If it's variable, it could indicate sensory feedback loops.
I think looking at the results that we had, the results would vary a lot. And I think also it would be interesting to look at like the speed of the first attachments compared to like maybe more the last attachments. I do think there might be like what you're mentioning, the sensory feedback loop that, because they're not always wrapping at the same latency time, right? And it does, I mean, when we looked at our result, there was a lot of variability. So yeah, I do think maybe it'd be interesting to look at the different recruitments and the time compared to like the first ones and the second or third one to the fifth one.
Mm-hmm. And then maybe a last one. Did I get it correctly that the latency time between suckers in the medial part is shorter than in the distal part? So there's faster sucker attachment in the medial part and slower in the distal?
The opposite. Okay. So it's faster in the distal region than in the medial region, which I think might just be because also we had way more samples with the distal, and like I said, this experimental design favored this distal recruitment, and also the fact that the neural segments in the medial region are a bit longer than in the distal region.
Mm-hmm. Have you tried to see whether there is a correlation between the latency time and the disc diameter? Because the disc diameter gives you an estimate of the distance between two diameters, yes. and if it's a fixed nerve velocity.
We didn't look at that, but it was recommended by someone in the lab when we were talking about the results, and this is someone who studies the nervous system of cephalopods. But no, it's not something I looked into, but it was mentioned as a good thing. That's why we had a ruler, and we were like, the only thing is that we didn't always have a very clear view on the sucker and to have a full measurement. But that is absolutely something that would be interesting.
Yeah, because it could also tell you something about, is the motor pattern fixed or is it modulated?
Yeah.
Okay, thank you.
Thank you, Dominique. Stina.
Yeah, thank you, Raphael. That was a really, really interesting talk. Completely out of my comfort zone. I've never even thought about this. I have a couple of questions. I think you mentioned that you said your sample size was really small, but maybe you mentioned it, but I don't think I've seen the sample size. So what was your sample size and how did that impact your analysis?
So the sample size is the amount of good clips that we had. They were different compared. So we had more in the distal region. So we had between 85 and 109, but then in the medial region, for example, in the direction towards the body, we only had 17 good clips, right? So this is where I'm like, okay, the meteor results are are a bit more less significance in terms of like stat significance because they were pretty low. And like I mentioned, it would be nice to have more of a standardized amount of clips so we could have more even statistics.
Okay, thank you. And because I don't know anything about octopus and their arms and their suckers, so does every arm have the same amount of sucker, and then does every arm and the suckers in the same positions have the same function?
So the function of each, well, I do believe, I think every arm does have about the same amount of suckers, and the function of each sucker is kind of a mystery and kind of what I was hinting to as maybe a hypothesis for that could be looked into is that they use different sections of the arm for different roles. Because I do, like they would bring food, like, and the proximal sucker, so the suckers closer to the mouth seem to like be the one kind of like handing the food or like different. So there isn't any necessarily existing literature proof on the differences between the suckers, maybe the different functions between them. But I do, from what I have observed, I do believe there is a difference in the way they can manipulate objects.
Yeah, awesome. So, and then obviously I understand the biotech context of your work and the ongoing project, but could you put this also maybe in an ecological context? Why is it important for us on an ecological level to understand the function of the suckers and the arms?
Yeah, well, I think knowing more about a species, like just fundamental research as a whole kind of makes you appreciate a species a bit more also, and understand its role in the ecosystem also, because if you know, I don't know, like they use more the distal for the fetching motion, so you understand, okay, well, that kind of correlates with the kind of prey they prey on, or the way they behave in the wild. But I do think on a more personal level, I do think that working with octopuses kind of, it made me see them in a very different way, where I had eaten octopus before and now I don't think I was. So, but that's very personal also. But yeah, I do think understanding more fundamental science on a species can just kind of break certain stigmas also around it. Yeah.
Yeah. Awesome. Thank you so much. And I'm really sorry, but I have to leave because I have to catch a train. And I just want to say thank you very much for having me today. It was super interesting. All four of you did a really, really, really great job. And I think I'm going to read one or two of your reports. So I'll look forward to that. And Merry Christmas and Happy New Year.
Now we're getting back to you.
I'm also kind of with Stina on this. It's out of my depth, so my questions will be quite general. Something that you just mentioned actually on the behavior, because you're only testing one behavior here, which is what you interpret as predation.
No, sorry. We didn't interpret it as predation necessarily. I was maybe just speaking in a more of a general level of like knowing how they use certain regions of the arm maybe for a certain action, could understand how they behave in their environment. What we were testing wasn't to mimic predation, it was purely just to have them recruit their suckers.
Yeah, so the motivation for it was wrapping motion. Yeah. So how do you think this would change if you were to assess different behaviors, if they were walking along a place or if they were camouflage, whatever?
Well, I do think that the experimental design that we had highly favors a certain, so the discs were against the wall, which obviously can impact the way they recruit the suckers versus like if they're in open water and they're interacting with an object that has way more texture. The original goal of this project was to look at how they recruit their suckers and objects with varying textures. So we've tested a lot of different things. We've tested with a live crab and everything, but it's really hard to have a clear view on their suckers, which is why we ended up with this experimental design. But I do think they would react differently and recruit their suckers differently in the wild or in a different environment. Like I do think there would be less strictly ipsilateral recruitments if they were interacting with an object that had way more texture or wasn't.
Yeah, there should be, because it's flat, right?
Yeah.
So there's a bigger surface area for all their suckers to go down, which would lead to this pattern. But in reality, where they would do this, some of the suckers probably wouldn't even recruit, I guess, on it. But it's
what we saw, we only took sequences which were, most of the sequences were that would happen in a row, right? But very rarely we would see that they would jump a sucker, for example, which I think would be observed more frequently if we were using an object with different texture and that wasn't as flat, right?
Yeah. And I don't think it's an easy thing to present, but I think you did a good job. The first and the last part where you talk about the soft robotics.
Yeah.
We make enemies here. I don't think it's the best way to learn necessarily, but because there's so many limitations to what the octopus can do and what maybe we want soft robotics to achieve. So my question to you is, do you believe in it? Do you think this can really help? Or are we just
not soft robotics? Help.
If
we can help. Yeah.
For soft robotics.
Oh, okay. It's
help for soft.
I do believe that if you're making a robot that you want it to do this exact motion, you're taking what the octopus would. So what we're looking at is what would be the most optimal way that the octopus is recruiting.
Mm-hmm.
So if you want a robot that wraps around an object like this that's against a wall, same design, I do think it would be very valuable. But obviously, if you want to make a robot that, like, I don't know, like grabs objects and serves you coffee or something, maybe this study wouldn't necessarily be as relevant. But when we think of soft robotics, maybe we think more of like a complex system, but it can also be something very simple as much as just a wrapping motion.
All this.
Or
without suckers.
Without it is a type of structure.
He's not fun.
Oh, no. Sorry. Thank you very much. Enjoy.
That was the last time. We have a few questions. All right. Yeah, you can pick your question.
Yeah,
I thought you did a really good job at explaining this complex study. But I don't kind of building off of what Dominique said. Did you just observe, maybe not statistically, a pattern of how an octopus would initially approach an object versus how it would utilize its suckers after a secondary attachment? Or
wait, what do you mean by secondary attachment?
So like its first approach when it wrapped around the disc, whether or not it was different from the second or thereafter when it's interested in this object.
So the first approach, I would say that a lot of the times you would see kind of this like, it would like, ooh, you kind of like point one of the suckers and then wrap. I'm not sure if they understand by the after they've already interacted with it. So you mean like if they were to, okay, maybe wrap around one disc and then they wrap around another disc in the same trial?
Yeah.
I think the way they approached the suckers, the discs were also different according to different regions of the arm. So the distal region would kind of do this thing, whereas the medial region was a bit more messy, I would say. Based on my observations, there are a lot of different ways that they approach the discs, and it's hard to have one clear conclusion because they had a lot of different quirks of ways of interacting with these discs. So yeah, but it would be an interesting thing to look at, the way they approach it. Absolutely.
Thank you, Rob, for your presentation. I just had a question. Do you know, and then how could we test this? Like, do octopus, they have like types of like soft and like hard grabbing? Like, can they like grab just like things softly, or when they need to really grab it strong? Do you think like you could test this? And
so that's really interesting because octopuses are really flexible and soft bodied, but they can also be really strong and rigid because they have really dense muscle tissues, which is what makes them muscular hydrostats. So it's like something that's really that has no structure like a skeleton, but is able to have like that stiffness. So I do see like the way they would maybe interact with things. Sometimes they would interact with it in a more of like like they can touch something without grabbing it, right? But then they can also touch something and you can see that they have a very strong grasp on it. That would be something interesting to study. I'm trying to think of how you would do that, and that would take a lot of brainstorming. But I do know that there is also a potential study in like measuring the different weights of like how they react to different weights, maybe different textures of the object, but also varying weights of objects.
Because I was thinking maybe the patterns also like is what is lateral and like the contrary, like how it will vary depending on the strength they need to grab something. And that can also give you a clue on exactly the patterns you need to make this soft robotics. So how hard or how soft you want things to grab.
That would be interesting. Yeah, like I could think of maybe like if they're trying to grab something that's like maybe heavier or harder, like if they're forcing on something, maybe there wouldn't be that ipsilateral one, there'd be maybe more of a cross route. But also I think of their suckers and how strong they are, and like working with them, sometimes they would like attach to you and like they stick, and like so I do think that even just the ipsilateral recruitment, just having a certain number of suckers, also the more suckers, the stronger grasp, right? So that's also something that could be looked at.
We learned from Gaia that the octopus have very different personalities and they're different individuals. You had 5, I think you said. Do you think the difference in the individuals would affect your results, or is this something that's very standardized in every?
So I do believe to a certain degree. It's not something we accounted for in the study. It was an assumption that there wouldn't be individual differences. I know there's another study that was recently published from this lab that they ended up looking at the individual differences just to make sure that there wasn't that effect, and there was no significance at all. I did see like certain octopuses had a certain way of grabbing things, like they do kind of like an inchworm, whereas like another octopus would never do that. But I do think that the certain patterns that were pretty like frequent. Like we did see a lot of ipsilateral across all individuals, but that was obviously an assumption of the paper of there wasn't that individual effect.
About just very quick, the recruit. You showed the different options, and it was always either left or right. It was never both.
No.
That's so interesting.
No. Yeah.
Do you have any what that could be?
It was never both. Sometimes it was a matter of a fraction of a second because we were looking at them like frame by frame by frame. So sometimes it was like I said that one attached first, and then my colleague was like, "No, I think that one's attached first," and then we go to your supervisor and we're like, So it's something we didn't account for, and we choose to put them in a certain order because that would be a whole other thing to look at. But there was always like, a matter of a frame.
So at the end of the frame, for example, all of the suckers would be attached, or only the ones that you marked? So there would always be.
What do you mean exactly?
When you say, okay, it was ipsilateral all the way through, and then maybe it crossed at the end.
Yeah.
What would happen to the other suckers at the same time? Would they just stay loose for the whole time?
Oh, okay. Yeah. So we went the first 5 suckers that attached.
Okay. And so after that, usually the rest would attach as well, or just like by experience?
We went, okay, for example, like we were, okay, blah, 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 this attached, this sucker attached. Now we're looking starting from this sucker, which is the next to attach afterwards. So maybe others did recruit here, but it's not what we chose to look at. But that's why it gets really tricky to quantify these kind of recruitments because we need to have a certain naming convention and a grading convention that needs to be agreed on, and the way you want to analyze it and what you want to take out of it, right? So we wanted to look at in what order do they go when they go in a certain direction, right? So we weren't necessarily looking at just like, okay, like all the suckers, like how do they attach, right? Yeah.
Thank you.
Thank you very much. Now I can push on this button. I wanted just to say congratulations to all of you. It has been a very, very good moments for us, and I think for you. You had a lot of classes, so I'm talking to the year twos here, and you had a lot of classes in this last 1 year and a half. It may be the last time you're sitting in this chair in this room, and I think you learn at least as much in 3 days with each of you than with all the teachers you had in this 1 year and a half. I don't know, maybe. So it was very nice. Also, I would like to thank all the jury members and reviewers. There are 20 people who took their time this week, but also during the Christmas break. You read your papers, and thank you for that. Alors, Sina, for example, is coming back with the Blue Manager in Bluefish. Dominic is coming back with the year one semester with the hackathon that apparently you asked really for it to come back. So the year one will have it with Dominic. Thank you for coming back. And Juliette, of course, is coming back because we love Juliette and Polmer for talking about aquaculture, about fish, about biotechnology. And also when it's time to take a bus and go for discovering startups and stuff like that. So I hope you enjoyed these moments. I wanted to once again thank you for really playing with what we proposed you to do. So most of you were here even early in the morning, and also during the coaching you were here for working together, and it's really something we appreciate. Enjoy the time. Of course, this afternoon we're coming back at 1:30. Yes, Christelle? Yep. Oh, good. 1:30 this afternoon for the big party at Lérins. 4 groups, and we learn a lot of things with Lérins. It's good. Okay. We're still live. It's fine. I haven't said anything. Yeah. Good. Dominic, Juliette, if you can wait for a minute, we will nicely ask the students for the last time. Leave.
Happy New Year, everyone.